Hello everyone. This is double helix structure of DNA, that is deoxyribonucleic acid, which was given by Watson and Crick. First we come to what is nucleic acid. Nucleic acids are the biopolymers of nucleotide. Which link together by phosphodiester bond. There are there are two kinds of nucleic acid. One is DNA, as we have discussed, and second is RNA, that is DNA is deoxyribonucleic acid, nucleic acid, and RNA is ribonucleic acid. As DNA is found in nuclei, mitochondria, and chloroplast, while RNA are found in ribosomes, and cytoplasm. Now we come to where they are located. DNA is present in both plants and animals. Whereas DNA or RNA present in Virus. Viruses may have DNA or RNA as their genetic material. Now, now composition of nucleic acid, which is very important. As nucleic acid is composed of sugars, nitrogenous bases, and phosphoric acid, in this sugars and nitrogenous base combine to form nucleoside, and when phosphoric acid or H3PO4 binds to nucleoside, they become nucleotide. These now these biopolymers of nucleotide is known as nucleic acid. Now as we come to the composition, first is sugars. In DNA, there is deoxyribose sugar, and in RNA, there is ribose sugar. The difference between the difference between The difference between the two is this 2 prime and that is which is not hydrated. As in RNA you can see that it is H and OH on the 2 prime and, but in DNA it is deoxygenated, as the name indicates, deoxyribonucleic acid. Now we come to second, that is phosphoric acid. Which contains 3 monovalent hydroxyl group and 1 divalent oxygen atom. As you can see, there are 3 monovalent hydroxyl atoms and 1 divalent oxygen atom. Third one, which is very important, is nitrogen bases. Which is present in DNA. There is a major difference between the nitrogen bases in DNA and RNA. DNA contains adenine, guanine, cytosine, and thymine. Whereas RNA contains adenine, guanine, cytosine, and uracil. So the difference between the nitrogen bases in DNA and RNA is the basic is thymine and uracil. In DNA, the fourth nitrogenous base is thymine, and RNA there is uracil in the place of thymine. There is always DNA is in the double stranded structure and RNA is in the single stranded structure. Now as we come to the structure, to the structure of nucleic acid, as I have already told you, nucleic acid is made up of phosphoric acid minus pentose sugar minus nitrogenous base. Nucleic acid is composed of phosphoric acid, pentose sugar, and nitrogenous base, and in this phosphoric acid and pentose sugar combines to form nucleoside, and H3PO4 combines to form nucleotide. The chain of these nucleotide combinedly form nucleic acids together. Now, as we form the DNA structure or polyribonucleotide structure, we first see there is a base, that is nitrogenous base. This nitrogenous base forms a glycosidic bond with the sugar, and here the phosphate group is attached. This phosphate group binds to another sugar Now this phosphate group is attached now you can see that this phosphate group is attached with two sugars, so this bond is known as, or this linkage is known as phosphodiester linkage, and the bond between the nitrogenous base and the sugar is known as beta glycosidic bond. The bases is when attached with the another base, then the bond is known as hydrogen base, and this whole polyribonucleotide is basically a single stranded molecule, as this is RNA. Now we come to the structure of DNA again. As you know, this is the structure we have already studied. This is the structure of DNA. The diameter of DNA is 20 angstrom, and the one complete turn of DNA is of 34 angstrom. The distance between one base pair is 3.4 angstrom. There are total 10 base pairs present, which combinedly forms 34 angstrom in length. Now some major points about DNA is: first point, it consists of two helical polypeptide chains. Which are coiled around a common axis. Second is, they both are anti-parallel to each other. They both are complementary to each other. Phosphate and deoxyribose are present at periphery. And plane of sugar is in the plane of axis, but plane of bases are perpendicular to each other, to both axis in sugar. Coiling of both the helices are plectonymic. As one runs in 3' to 5' direction and another runs to 5' to 3' direction, so these are anti-parallel to each other.